Geef een applaus.
Welcome, everyone. Hereby I open this academic ceremony in which Irene Hemel will defend the academic thesis, Imaging Mitochondria from Dynamics to Brain. May I invite you to present a summary of your study and the conclusions of your thesis?
Thank you, Pro-Rector. Dear members of the Corona, dear colleagues, friends and family, thank you all very much for taking the time to be here today for the defense of my thesis. And before I go into my research, let's take a moment to appreciate something remarkable. Ourselves. Every single individual in this room is composed of around 30 trillion individual cells. And each of these cells is a highly organized compartment with different structures working together to keep things running smoothly. At the heart of it all are the mitochondria, tiny structures with massive responsibilities. They are, for instance, important in programmed cell death and the production of heat, but most notably, they produce the cellular energy that keeps us alive. They may be microscopic, but without them, none of us would be here today. When mitochondria don't function properly, a decrease in the amount of energy production is a direct consequence. And while this is important for many processes, in humans, it's most commonly visible in the muscles and in the brain, as these tissues consume the most energy. What kind of diseases am I then talking about? Well, there's mitochondrial disorders, where proteins inside of the mitochondria don't function properly, directly influencing the amount of energy that can be produced. But mitochondria are also known to not function properly in neurodegenerative disorders, such as Alzheimer's or Parkinson's, as well as various types of cancer. Luckily, mitochondria have several mechanisms to keep themselves more healthy. And one of these is mitochondrial dynamics. And mitochondria can change their shape based on what the cell needs. In mitochondrial fusion, two separate mitochondria are combined into one larger structure. And during mitochondrial division, one larger mitochondrion is separated into two or more individual pieces. For this to happen, first, a preconstriction of the mitochondria happens, caused by other structures in the cell, before a final cut is made, causing it to separate into individual structures. This is, for instance, important to split off damaged parts of the mitochondria so that they can be recycled, and the remaining parts of the mitochondria in the cell are more healthy. Considering the amount of processes that mitochondria are important for, as well as the detrimental effects of their dysfunction, it's important to study how they work. This can be done with several different kinds of techniques. You can, for instance, use laboratory-based experiments, where you can measure how much energy is being produced, or how much of a specific protein is present in the cell. Additionally, you can use imaging-based approaches, where you can see how the mitochondria are distributed in a cell, or how they behave. And you can use computational-based approaches, where you can model and predict how mitochondria function in specific situations. One of these individually is not going to give you a complete understanding of how mitochondria work. But by combining different approaches, you can gain a more comprehensive understanding of mitochondrial functioning. If we look at imaging a bit more closely, imaging of mitochondria can actually be done at multiple different levels. Electron microscopy, for instance, can be used to image individual mitochondria. And here you can even see their inside structure. A downside of this approach is that you can only image a very small part of the cell. At a cellular level, you can see how mitochondria are distributed and how they move, but you lose detail that you could see on the mitochondrial level. At a tissue level, you again lose information on the individual mitochondria, but you gain information on how cells work together, which is more representative of what's happening inside our bodies. and techniques like PET or MRI can even be used to image entire organs. And here you can see the effect of mitochondrial dysfunction on, for instance, the brain. In my thesis, we combined imaging at several of these levels with laboratory and computational-based approaches to answer various questions about mitochondrial functioning. In Chapter 3, we tried to identify new proteins that are involved in the dynamic behavior of mitochondria. And for this, we first predicted which proteins might be involved, and then selected several candidates that we thought were interesting enough for laboratory-based validation. The first step of this validation was to decrease the amount of this protein that was present in the cell and assess whether this causes a change in the shape of the mitochondria. We used microscopy to take images of these situations, but just taking these images is not enough. We needed an objective way to prove whether there was a difference in mitochondrial shape between the control condition and a condition in one of our proteins was decreased. Therefore, we assessed 5 different mitochondrial quantification tools, and all of them follow the same basic principle. If you have an image of mitochondria, here represented by the schematic drawing, the first step is to set a threshold. And the threshold determines what this program recognizes as mitochondrial signal and what it considers to be background. Imagine you have a black and white photo. A threshold is like saying everything above this level will be considered as white, and everything below it we will consider as black. In an ideal situation, this threshold will be set at a level that all of the mitochondrial signal is taken along without anything else in the image. This thresholded image is then analyzed, and an output is generated containing information on the number of mitochondria, their size, and several other parameters. If you do this on drawn mitochondrial networks, as you can see here, all 5 of these tools work similarly well. But real microscopy images have imperfections, and this is where we saw a difference. 4 of the tools use one threshold level across the entire image and took along a lot of the background signal in the thresholding step, resulting in an inaccurate quantification. Only one of them changed the threshold level across the image and was able to properly separate the mitochondrial signal from the background. We then used this tool to assess whether there was a difference in our images and found that a decrease of several of our proteins of interest caused an increase in average mitochondrial shape. The protein for which this increase was the largest was studied further. For this protein, ITP-RIPL2, we assessed where it was present in the cell. And to our surprise, this was not in the mitochondria, but it's instead present at a cell support structure. Additionally, if we decrease this protein, this cell support structure is completely absent. This tells us that mitochondrial shape is influenced by factors outside of the mitochondria as well, and that it is important to take this along when considering their dynamic behavior. Additionally, while we were making the images for the quantifications, we found something unexpected. Normally, if you decrease the amount of division that happens in the mitochondria, you get long and interconnected shapes. But in the cells that we were using, fibroblasts, we found a different phenotype, which we call the pearl necklace mitochondria, where bigger blobs of mitochondria are connected by only tiny pieces of mitochondrial string. Follow-up imaging studies showed us that this phenotype is actually the result of light exposure during microscopy. Microscopes that use more light to visualize the mitochondria resulted in a faster generation of this phenotype, and microscopes that used less light, this phenotype came up at a much slower pace. We were then interested to see what this phenotype looks like in more detail, and generated a 3D model of the mitochondrial structure using electron tomography. In this model, you can again see the bigger blobs of mitochondria connected by only tiny tubules. In these bigger blobs, here seen on the right, the mitochondria kind of seem to be pulled apart, and their inner structure is disrupted. At the smaller tubules, we found that filaments were present. And the presence of these filaments can point towards a mitochondrial preconstruction. A further discussion on how these filaments could be involved in preconstruction of the mitochondria was discussed in Chapter 4 of this thesis. At this point, we're already pretty far zoomed into the mitochondria, but if you could look even closer, you would see that they actually have their own DNA. And this DNA is essential for their functioning. For a proper maintenance of this DNA, it needs to be multiplied. And for that, the protein POLG is extremely important. Humans have 2 copies of POLG encoded into their DNA. If you have 2 healthy copies, you have a sufficient amount of energy production and a happy, healthy brain. If someone has 2 broken copies of POLG, the amount of energy that your cells can produce is too little, and you get severe neurological phenotypes, often with detrimental consequences. Unfortunately, we still don't really know how this disease develops exactly, and if there are specific regions of the brain that are more vulnerable to a decrease in energy production. Therefore, we looked at individuals that have 1 healthy and 1 broken copy of POLG. And while these individuals are not sick, we believed that they might have a tiny bit lower energy production than individuals with 2 healthy copies. And we were then wondering if we could use these individuals to find minor changes in their brains and identify vulnerable brain regions. How did we do that? We compared individuals with 2 healthy copies to individuals with 1 broken copy of POLG using 7 Tesla MRI. What does this 7 Tesla mean? Basically just that this MRI machine uses a stronger magnet than the ones that you might be used to from the hospital. And because of this stronger magnet, we're able to visualize the brain in more detail and can thus find smaller changes. Analysis of this data and comparison between the 2 groups showed us that there were no changes in brain structure, but we did find a mildly decreased blood flow in individuals with 1 broken copy. This decrease in blood flow was also present in brain regions that are commonly affected in patients with 2 broken copies of POLG. And a different study found a decreased blood flow in similar regions for a different mitochondrial disease. And this tells us that these brain regions might indeed be particularly vulnerable to a decrease in energy production. In summary, my thesis has shown that selecting the right methodology is important for an objective quantification of the mitochondria, that structures outside of the mitochondria are important to maintain their mitochondrial shape, that it is important to always check for mitochondrial artifacts when imaging living cells, and that mild changes in blood flow seem to be present in the brains of individuals with 1 broken copy of POLG. And while we are still far away from completely understanding how these complex dynamic organelles work, we believe that imaging them at multiple levels will be necessary to gain a completer understanding of how mitochondria function in healthy and diseased situations. And with that, I would like to thank you for your attention and give the word back to the Prorector.
Thank you very much for your very nice and clear presentation. Before we move to the discussion, let me first use the opportunity to introduce the supervisory team for this thesis. There's Professor Arts, Dr. Gerards, and Dr. Morel. And according to Maastricht tradition, they will not be asking questions today, so you might otherwise wonder why are they here and why they're not saying anything. My name is Ralf Peters. I'll be acting as the pro-rector, meaning I replace the rector in this capacity, and I will be guiding the procedure. And that brings us to the opposition for this defense. The opposition will be opened by Professor Roebroek. Professor Roebroek is a professor of brain connectivity and computation at this university. He was chair of the assessment committee and is also acting as today's secretary to the degree committee. Please, Professor Roebroek.
Thank you, Mr. Prorector. Dear candidate, First, I'd like to congratulate you on a beautiful work and a beautiful thesis, which you just very nicely summarized. I was particularly impressed, and so were other members of the committee, by the great many techniques by which you try to answer a question. And in the true sense, I think, of system biology, going over multiple scales and multiple measurement modalities, ranging from, as you showed, electron microscopy to optical microscopy to MRI. So well done to you and to your supervisors. I also want to extend those congratulations to your parents and your family and your friends. Of course, I'm also here to ask a question here and there. My specialty is actually MRI, although I also do quite some microscopy, but there are microscopy experts here that will ask questions about that. And so I want to focus on chapter 6, which you just very nicely summarized, where you did 7 Tesla MRI of monoallelic carriers of the Pol G mutation. Now you showed that you actually detected differences in blood flow in some areas, but not others. You mentioned in the presentation that these largely seem to be the same as those that are affected in full carriers and that have pathological symptoms. From the thesis, I also got it that there are some differences, like the occipital area. Can you first answer for me your best theory and speculation even, with arguments, why it would be these areas that are affected more than others in the brain? Are they more vulnerable? Do they need more blood flow? Do they have a different architecture? A lot of the cortex is similar, but a little bit different. Can you tell me what your thoughts are about this?
Highly esteemed opponent, thank you for your kind words and your question. I do indeed think that it might have something to do with maybe the distribution across the cortex. So it's recently actually been found that the mitochondrial, the amount of mitochondria that's present across the cortex also differs greatly. So that in specific regions of the cortex there's more mitochondria present than in others. So that must mean that there is a difference in what is needed across the cortex as far as energy production is concerned.
Were those areas that you found that were found to have more mitochondria?
I'm not sure. The article came out 3 days ago.
That's short, yeah. All right, I want to follow up with a more technical question about MRI, obviously, which is a technique that I love, but the methodological details are not so important today. Diffusion MRI, or DTI, diffusion tensor imaging, is a technique that applies a certain type of contrast on the machine to look at how basically molecules move, either through diffusion or to blood flow. You apply DTI. I don't believe you've reported on the DTI, but you can correct me if I'm wrong. But there's another diffusion MRI technique called intravoxel incoherent motion imaging, IVIM imaging, that is actually quite exquisitely sensitive to flow in very small capillaries, so let's say in the microvasculature of the cortex. Now you found your differences in the ASL technique, which is more sensitive to the macrovasculature and maybe the mesovasculature. So can you tell me, one, why you chose not to apply IVIM? And if there wasn't a lot of thinking about that, and can you rather from me speculate that if you both used ASL and IVIM in a follow-up study, what the different sensitivity to, let's say, macrovasculature versus microvasculature might give for this particular disease pattern?
Yes. So when the study was designed, we had absolutely no idea where to find or expect any potential differences. And we decided to set it up in a way that there was a broad amount of scans, so that if there were differences, we would be able to pick them up some in one of the scans. We actually were not able to analyze the diffusion-weighted imaging at this point. This is also why the results were not reported yet. That's still planned for the future. But basically, that's the reason why probably this was not in there, because we wanted to look at a broad range of different types of scans. I can imagine, though, that if you have information on the microvasculature as well, that you can get a more, I guess, detailed map of where exactly the blood flow might be different, because now the areas that we found are still pretty large, and we also analyzed it in a way that we can find pretty large areas. But I would also be interested if it's like the amount of blood flow to the capillaries would be necessary, or if that's also decreased, or if it's just like a more general pattern that in the bigger blood vessels this level is decreased.
What's your thought as a final short question? Would it be or wouldn't it be?
I think it would be.
Interesting. Well, thank you very much for your answers. I'll give the word back to the prorector.
Thank you, Professor Roelbroek. And the opposition will be continued by Professor Jacobs. He was a member of the assessment committee, and he's Professor Emeritus of Molecular Biology at Tampere University in Finland. Please, Professor Jacobs.
Thank you very much. And I'd like to open my questions and comments with seconding the congratulations offered by my colleague to you, Irene, as the candidate in this thesis dissertation. I also found it a really stimulating, interesting, and in many places challenging piece of work, and I think you can be really proud of it. And since I assume nobody else is going to mention it, I'd like to also congratulate the designer of the image of your thesis book, which you're showing on the screen, which is a wonderful summary of the entire thesis. And I guess for members of the audience here who are not molecular biologists, just looking at that, they would probably, at least if they were told that the image on the right-hand side is that of the mitochondrion, I think to most of you it looks a little bit like a sort of baby in a crib. And it kind of nicely symbolizes the fact that, at least for those of us who do work on mitochondria are our baby. I'd like to start the questions by referring to your propositions that you issued with the thesis, which are challenging and very strident. And I'd like to focus initially on number 6. So if we can read out that proposition.
In order to fully understand mitochondrial biology, it is imperative to look beyond mitochondria themselves.
Thank you. So my question is, can you give us an example from your thesis work that actually illustrates that point? And part B of my question is, can you go beyond that and cite something else in mitochondrial biology that isn't part of your thesis to which that applies? So, looking beyond mitochondria.
Dear highly esteemed opponent, thank you for your kind words and your questions. Yes, I can absolutely name an example for my thesis and beyond. So in my thesis, we've looked at the intermediate filaments as a part of the cytoskeleton that are important or seem to be important for mitochondrial fission, as well as the ER, which is known to be involved in mitochondrial fission. But the other cytoskeleton components, like the microtubules and actin filaments, are also very important for the maintenance of the mitochondrial morphology.
Yep. And beyond your thesis?
Beyond the thesis. Well, other cytoskeletons are beyond the thesis. There's also a large interaction with endosomes and going into eventually liposomes and degradation of the mitochondria, so to recycle the damaged compartments.
So that mitochondria are not just floating in outer space, but they are in intimate communication with the other components of the cell, and we have to try to understand them not merely in terms of what they do as isolated organelles, but as part of a community. Can we also learn something about their biology by looking at their ancestors in aerobic bacteria?
Absolutely. The entire theory of where mitochondria come from is that they are from a bacteria. And I know that the way that energy is produced, that there's other, like, I guess, backup mechanisms in bacteria still present that are not present in humans anymore. So trying to understand how those backup mechanisms work and how they might be able to be altered or induced in humans could help us understand or even maybe treat mitochondrial dysfunction as well.
Okay, I'd like to turn to some more sort of specific scientific issues arising from your thesis. So one of them that really struck me is your discovery that a protein that's been implicated in the assembly of oxphos complex 1 is involved in mitochondrial dynamics. And you suggest that the hyperfusion that results from its absence is probably due to a stress response to a defect in complex 1. And there are some other reports in the literature where inhibitors and other insults on complex 1 can produce that result. But I'd like maybe to ask your speculation on a deeper level of why what is it about complex 1 that provokes that response, and how does mitochondrial hyperfusion actually assist the processes that mitochondria have to undergo that would be dependent on complex 1 specifically?
So the idea behind this hyperfusion in response to stress is that the cristae can be more abundant if the mitochondria are larger, because the membranes, there's more space for the membranes inside of the larger mitochondria. And I know that complex 1 is extremely important, and especially in combination with actually as supercomplexes with other complexes together to maintain this cristae shape. And then, in theory, the more cristae they are, the more space there is for the oxphos complexes, and the more energy that can be produced. But we also know that this is only temporarily, and that in the long term, this hyperfused state is not well maintained for an prolonged enhancement of the energy production.
Does interference with other oxphos complexes produce the same result?
I believe that interference with complex 4 would do something similar, but most of what I've read is for complex 1 indeed.
Right. But on the other hand, we also observe that when the mitochondrial membrane potential is disrupted, which is a really severe disruption of mitochondrial function, the very opposite happens. Instead of getting hyperfusion, we have massive fragmentation of the mitochondrial network. So how do you explain the fact that different stresses are producing actually opposite effects on mitochondrial morphology?
I think it would depend on the excess of the stressor and how well the cell might be able to still compensate for or to deal with the stressor. Because if the stress is too large, eventually cells will go into apoptosis, and for that, the massive fragmentation is necessary. So it could be that the disruption of the membrane potential is just so detrimental immediately that they go towards the fragmentation and apoptosis site, where a decrease in complex 1 can be temporarily compensated for, and then if the stressors disappear, that the cells can recover.
So do you think that the hyperfusion response is actually part of a mechanism to restrain apoptosis and those processes that would lead to cell death?
Yes, I think it might be.
Okay, I'm not quite sure how much time I have left, so.
If you have a very brief question with a brief answer.
Okay, maybe I'll just ask a brief question, which is, when we come back to the pearl necklace observation, which you bravely drove forward and basically shown that this really interesting morphology that you got was an artifact. Beautiful artifact, nevertheless. But the question is, what was it about the work that actually led you to believe that it was an artifact? I think a lot of people would have just published that finding. It's a pretty picture, and it looks like it's something really profound that's going on in mitochondria, and yet you were convinced that it was an artifact and wanted to drive forward, at least ask the question clearly. So what was it about the observation that led you to believe that?
A coincidence. So we tried the electron microscopy And we couldn't find anything in the initial phases, and then we were like, okay, maybe our knockdown didn't work. And then we decided to test that before fixing the cells. We happened to use a different microscope, and it was like, okay, this is happening. And then from there, we started to look at what might be causing it.
I think that was very brave, because most people, I think, would assume that it was the electron microscope that introduced the artifacts, rather than the light microscopy. Thank you very much.
Thank you, Professor Jacobs. The opposition will now be continued by Dr. Carlier. She was a member of the assessment committee, and she's an associate professor at Merlin here at Maastricht University.
Yeah, thank you very much. Dear candidate, also I would like to congratulate you. I think it was a very nice thesis, which indeed involved many interdisciplinary aspects. It was very well organized and very methodical. And I also want to congratulate you on the very nice out-of-the-box chapter titles that were very interesting and very nice schematic figures. But of course, I also have some questions, and specifically I would like to focus on chapter 2, on the algorithms, which you also shortly explained in your presentation, to analyze the images and to quantify them. And then specifically, for example, in figure 1 and table 1, you use actually drawings in order to test and compare these different quantification tools. But yeah, they do not seem all to work equally accurately, and you explain this from the thresholding algorithm, but I find this actually a bit curious because your drawn images are actually binary. So how can then the thresholding be such an important explanation for this difference?
Dear esteemed opponent, thank you for your compliments and your question. The way we drew the images was just by hand, an imaging tool or Photoshop tool. And I think the points where it actually doesn't recognize it properly between the different tools were also the points that were the thinnest in the drawing. So it was, even to us when we were manually assessing them, like doubting whether or not these were connected, yes or no. So then the question is, did we manually assess it wrong, or did the tool assess it wrong? So I think it's more that the points in the drawing were so thin that that's what made it difficult to see if they were connected, yes or no.
Yeah, okay. Yeah, because you also mentioned indeed in your methodology that you use a block size of 10 and a C-value of 2. Or 10 and 5, depending on the type of image. So could you maybe just shortly explain what it actually means and why you changed these settings?
So the block size is indicative of how much of the image is considered for one threshold. So basically it just makes a lot of tiny thresholds across the entire image. And the block size is like how big one compartment is. And the C-value is a way of filtering out some of the background signal. And we found that the lower you go with the C-value, the more that was filtered out. And therefore, we also changed it between these drawn images and the actual cellular images, because the levels were different and the size of the mitochondria was different, because these are binary structures and the actual microscopy images have proper sizes attached to them.
Yeah, and so if you would have different types of figures, every time you need to kind of do this manual optimization procedure, or how did you optimize these settings?
Yes, so trial and error, mostly. Once you have the settings for a specific cell type, I found that it's fairly close to those settings, and we always adapt them for one whole experiment. So all conditions in one experiment are analyzed with the same settings. And then if we went from fibroblasts to HeLa cells, the changes in analyzing parameters were a bit bigger, because the size of the cells and the size of the mitochondria is different. But yeah, basically I have like a baseline of values. I try that, and I try some values around it to see if I can get it a little bit better. And then, yeah.
Yep. Okay. And then I would like to follow up on this a bit, because you also highlight this in your discussion, that the settings should be determined for every individual experiments for good reasons, because the conditions change and the background signal might be different. But still, yeah, I'm not sure whether I agree, or I would like to discuss with you, because isn't the idea in science that we design experiments in such a way that we keep most things under control in the same, and only change one factor, whereas now you have different cells, different settings, but also different settings to analyze the data. So how do you think this could influence your findings?
Yes, ideally you would be able to keep them the same. Unfortunately, while taking the images and doing the stainings, and they are still living cells, they're doing stuff, and I can control always how much background signal is going to be present in the cell. And if there's more background signal present, I will have to find a way to deal with that. Otherwise, the quantification will be way more off than when I do change the settings. I think that if you can manipulate the settings in such a way that you can get any answer out of these images you want very clearly, which is why I think you should. So basically we set the settings on the two extreme positive controls always. And if we can quantify both fragmented and elongated networks accurately, then all of the cells should be analyzed in a relatively well manner. And that should be pretty consistent across different experiments as well.
Okay, thank you very much. I hand the word back to the prorector.
Thank you, Dr. Carlier. Then the opposition will be continued by Dr. Spelbrink. Dr. Spelbrink was a member of the assessment committee, and he's a principal investigator at the Radboud University Medical Center in Nijmegen. Please, Dr. Spelbrink.
Thank you. Dear candidate, I would also like to extend my congratulations to you and your supervisory team for a very comprehensive and well-written thesis. So I would like to start the discussion talking about chapter 3. So you describe that the loss of intermediate filament integrity results in more interconnected and extended mitochondrial network. And you do some double knockdown experiments to show that it's rather inhibition of fission and not stimulation of fusion that causes this phenotype. So in the discussion you don't really speculate about how do intermediate filaments, how are they involved in fission? So perhaps you can speculate now.
Dear esteemed opponent, thank you for your kind words and your question. Yes, I don't think that the intermediate filaments are directly involved in mitochondrial fission, but rather that's an indirect effect that causes the change in the morphology. We know that the microtubules are important for the transport of the mitochondria. The intermediate filaments are more considered to be like anchoring points for mitochondria across the cell. So that one of the things it might be is that if these anchoring points are not there, the mitochondrial shape cannot be properly maintained across the cell. And that the connections between the intermediate filaments and the microtubules are also necessary for the proper transport. And we believe that maybe if the transport cannot properly happen in the cell, then there's not really a need for the fission to take place as much, because they have to be smaller to be transported properly. And if the transport cannot happen properly, then why would the cell go through the hassle of creating these smaller fragments of mitochondria that can then not be transported anyway? Alternatively, the intermediate filaments are also, or have been shown to cause more of a bending of the mitochondria, where microtubules apparently prefer an extended mitochondrial network. So if you decrease the amount of the intermediate filaments that's present in the cell, and the microtubules are still present, and they prefer to have a more interconnected mitochondrial network alongside them, then that would also might be a reason.
Okay, thank you. So how would you test that hypothesis? Have you thought about experiments to test this?
Yes. How would I test that? I don't know if the cells could survive this, but I would be interested to see what happens if the microtubules are gone, and maybe even microtubules and intermediate filaments at the same time. But I don't know how well cells will tolerate completely messing up their cytoskeleton network to that extent.
Right.
And alternatively, to check if that's even the right direction, I would be interested to see in like where the known fission proteins are located in the cell and if they are disrupted, just to make sure that we're not looking in the completely wrong direction. And it's actually just a localization of the fission proteins.
Yeah.
It's more of a
Could live cell imaging tell you something
lifestyle
to look at the dynamics of the mitochondrial
Yeah.
network, since you...
Yes, if you could stain the different cytoskeleton or the intermediate filaments with live cell staining. I haven't found one.
Okay.
So I have looked for it, but I haven't found one that at least can be used as a dye. So you would have to make a fluorescently tagged version of the intermediate filaments. And I think that might be a bit problematic because they are like a lot of separate proteins that are composed into these filaments. And if you add fluorescent protein to each of these components in this network, it might be a bit difficult to assemble the network properly.
I understand.
So,
But you want to see the dynamics of the mitochondrial network as well, right?
yes,
So that
you would have to stain
could-
both of them at the
yeah,
same time.
yeah.
Yes.
Okay. And what about the interaction with the actin cytoskeleton and intermediate filaments? Is there any evidence for that?
Not that I've found, other than it also prefers mitochondria to be more bended and a bit shorter, similar to the intermediate filaments. And that the actin filaments are for sure very important for mitochondrial dynamics.
For fission?
For
also?
fission apps specifically, yes. But we did not find changes in the actin structure when we knocked down at least ITP.
Okay.
That was, I don't think it was in the thesis, but we did do the
right.
experiment. So the actin filaments did not really seem to be affected by this change in the intermediate filaments in our experiments.
Okay, thank you very much. Do I still have some time for a short question? Yes. So I was also triggered by some of your propositions. Perhaps we can start with number 5, if one of the paronyms can read it.
Proposition number 5, experimental work should always be guided by computational modeling.
Okay, thank you. So I guess my question is if you are suggesting that before we do any experiments, should we first go to the computer, make a model, and then start to think of an experiment? Or we can ask ChatGPT maybe what we should do. Or,
Well, I don't think asking ChatGPT is a very good idea.
No,
Because
I agree.
apparently fibroblasts don't have mitochondria, so.
Okay, that's
But
interesting.
I don't necessarily mean that you always need to start with computational modeling, but you can also use it along the way by if you generate like larger data sets and use them to further generate hypotheses. So I don't think it's necessarily starting with the modeling, but using it alongside the experiments, I believe can speed up some of the processes quite a bit and can also give you insights that you would not have gotten simply by doing the standard set of experiments that people are doing.
Right, yeah, I can agree with that. I would have almost turned the whole statement around that computational modeling should always be guided by experimental work, actually.
Also, yes, absolutely.
Okay, thank you. I can give the word back.
Thank you, Dr. Spelbrink. Then the opposition will be continued by Professor van Zandvoort. Professor van Zandvoort is a professor of advanced optical microscopy here at Maastricht University, but also in biophysiek der microscopie at the Aachen Universitätsklinikum. Please, Professor van Zandvoort.
Dear candidate, first of all, congratulations for you, your family and the promotion team. In my opinion, you wrote a very well-written and complete thesis, and I want to congratulate you with that. As a microscopist, as an optical microscopist, I specifically appreciated your first introduction chapter, which, in my opinion, that is meticulously and very completely describing what kind of imaging techniques are available for various types of tissues, like cells and tissues and organs. Now, I think this would be a perfect chapter for a course in urobioimaging describing how you can image mitochondria. However, nothing is without questions, of course, and without open issues. So I want to focus my questions on that introduction chapter, and especially, of course, where you describe optical microscopy. First, a general question. So you describe at various points in your chapter, quite a lot of points in your chapter actually, that laser scanning microscopy, whether it's confocal or super resolution or two-photon, has a disadvantage of slow speed. And that's the reason why you go, for example, to spinning disk confocal, but you could also do that for two-photon. However, you seem to forget that there's something like resonant scanning, laser scanning microscopy. And could you describe to me what would be the advantages and limitations of that type of very fast scanning in this kind of research?
Dear highly esteemed opponent, thank you for your kind words and your question. This is actually not a microscopy technique that I'm familiar with myself, but I can imagine that if you have a laser scanning confocal technique that can image very fast, that the effect of the phototoxicity might be lower, which would be definitely a huge advantage in imaging live cells or even living tissues. And especially then the imaging depth that you could get with the laser scanning confocal systems, you would in tissues maybe be able to image a bit deeper. I don't think for cells it would make that much of a difference because they're in a monolayer too little anyway.
Well, resonance scanning microscopy is available in Maastricht in the core lab. And it doesn't increase the penetration depth so much. Okay. That is not the point. But the point is that when you go quickly over a point where you have a certain intensity of your fluorescent marker, that it matters whether you stay 1 second on that point or 10 times 1/10 of a second. For the photobleaching, that is important. That means that when you go, for example, to your chapter on the pearl, that might have been a good option to look at and to at least include that in the analysis. Yeah? My next question is about your remark that you indicate that STAT phototoxicity depends on the specific properties of the laser. Can you extend on that? Because that sentence is not completely clear to me.
I think it's in general, but mostly the wavelength of the laser that would cause the difference in phototoxicity, where the lower wavelengths are generally known to have an increased level of phototoxicity than the higher wavelengths.
But you make this comment specifically about STAT microscopy.
This is indeed the case in general.
Yes, but your statement is specifically about STAT microscopy. What laser do you mean in STAT microscopy?
I know there's two. So then I'm trying to get the picture of the different lasers in my head. I think that the If I remember it correctly, but it's been a while, there's one in the middle and one more surrounding it.
Yeah.
And then I assume the one in the middle that actually causes the excitation would be the one where it's the most problematic. Now, apparently, no, the other one.
It's the other way around. So you have a laser that excites the molecules and a laser that depletes molecules. Yes. That makes sure that a large part of the molecules is not excited, or is excited but not fluorescent. And that last laser has a donut in the middle, has a hole in the middle, yes. And that makes sure that only in that small region where there's a hole, the fluorescence takes place. Now, that laser is the laser that, for example, in the microscope in Maastricht, has a very short wavelength, 592 nanometers. Do you think that if you would go to larger depletion laser wavelength, that the damage would be more or less? And basically, you've given the answer already.
Yeah, so I assume less.
Less. So we don't have that laser in Maastricht. So yes, for us, bleaching is a real problem. But if you would go to the modern STAT microscopes, like lasers 665, 675, 775, that would be much less, especially if you would combine it with resonance scanning. So I think there's still some options to work in STAT microscopy. My next question is a little bit later. You say storm is suitable for living cells. I hope you agree to disagree with me on that.
I've found some sources that say it would technically be possible, which is where the statement comes from, but that it's absolutely not ideal.
Why not?
Because it's very slow and a lot of images have to be taken to reconstruct the final version.
That's
of the
one
high
aspect,
resolution.
yes. You actually give the answer in the table on page 8. I think it's page 8, I hope at least. Yeah. If you go to table 1 and you see storm and you see the imaging depth.
Yes.
That's 200 nanometers. That means that you can only image on a flat surface of a cell.
Yeah.
And I see the prorector indicating that my time is over. So thank you.
Thank you, Professor van Zandvoort. And then the opposition will be continued by Dr. Koort. She's an associate professor at the Department of Translational Genomics here at Maastricht University. Please, Dr. Koort.
Thank you. Yeah, first of all, let me congratulate you as well of a wonderful thesis. It was a pleasure to read it. I think you went into really explore the dynamics of mitochondria in great detail, as already said, combining various techniques, laboratory experiments, combining with computational approaches, and in-depth imaging. So well done. I also want to extend my congratulations to your supervisory team. I have some questions about chapter 3. Maybe that doesn't surprise you. I think this is really a wonderful example that shows that computational approaches and laboratory experiments go hand in hand. And that's what you demonstrated there. You started with a protein-protein interaction network of known proteins in mitochondrial dynamics, extended that, and then finally you ended up with 4 candidates that weren't known yet in the mitochondrial dynamics. If you then knock them down in fibroblasts, you see indeed the morphology changes, and the one that showed the most severe change, you took that and went into more depth. And you said, well, it co-localizes with vitimentin, and then I want to see also with docking experiments whether they bind to each other. I think that's good, and you also see some 2 possible configurations there, but I'm wondering, couldn't you go back to your network analysis and also benefit from that approach in finding some ideas about how these 2 proteins could interact with each other?
Dear esteemed opponent, thank you for your kind words and your question. We have actually tried. So I did go back to the network and see if there was something that we could gain from that. But the amount of information that was available on this protein was very, very limited. We were actually surprised it even came up in the original analysis. We're glad it did. But, so there was also pretty much nothing known about other interactions that there might be, other than the few proteins that it interacts with in the network as well, one of which we also had as a candidate.
Yes, yeah, that's what I saw, because I checked and I saw that as well, that there is one link between...
Yes.
Okay.
And that protein, we didn't study that much further. We tried to look at the localization, but there were some experimental problems.
Okay. That's a pity.
Absolutely would have loved to see where this one is localized as well, but we couldn't find it.
Yeah, that was the MB2.
MB21D2, yes. Okay. And there's a third one, MAB21D3, I think, but that was only expressed in brain tissue, which made it very hard to do anything experimental with that.
Yes. Okay, thank you for clarifying that. Then you continue with the cells knocked down in the names ITPRIPL2 in two types of cells. And you did transcriptomics analysis and looked at pathway analysis. And then if you looked at changed genes, there's a difference. In the fibroblast, there are a lot of differentially expressed genes, whereas in your HeLa cells, it's less. And you say this is because of tissue-specific effects, and you also see that or cell-specific effects, and you also see that back in your results of your pathway analysis. Could you elaborate on that? Because it's difficult for me to see those cell-specific effects.
I don't mean the cell-specific effect in the effect on the mitochondria itself.
No.
Just the HeLa cells are cancer cells, so in general there is a lot of pathways that are dysregulated in these cells. But also because they're cancer cells, they mostly use glycolysis to produce the energy and are less dependent on the mitochondria for the OXPHOS. So that the expression patterns that might change might be less severe than they would be in fibroblasts that are more dependent on this.
Maybe going back to the start of the experiment, why did you choose to investigate the HeLa cells? Yeah.
Because they're very easy to culture and they're readily available in any lab.
Because I was wondering there as well, of course, if we look at how the abundance of mitochondria in cell types, that wouldn't be my first candidate cell, let's say, to check the
no, which is also why we decided to focus most of the experiments on the fibroblasts. But we did want to know whether this was something that might only occur in fibroblasts or if the effect that we were seeing was more spread out to at least one other cell type as well.
And one other thing I read in your chapter as well is that there was some problems occurring if you look at the effects in this HeLa cell with some time that they were frozen. So some were frozen for a longer period and then you needed to exclude one of the HeLa cells.
Yes, in the transcriptomics analysis. One of the data sets really showed a separate clustering from the other three. And we think something happened with the storing of the RNA, that it wasn't.
Okay, so it's really, and you're sure that that didn't happen in these other three cells that were left, and that could explain then maybe the lower amount of differential expression.
The other three were done later than the first one that showed the difference in expression pattern.
Good. You also conclude that actually the effects that you see in the knockdown probably are not going through expression, right? If you then look at the effects of the mitochondrial dynamics, so it's not about the expression level. If you then, so something else is happening, then probably how would you explore that? What kind of data, maybe other omics data, would you need in order to get a better picture of the mitochondrial dynamics?
I would definitely like to get data on a protein level. But I think it would need to be data on a protein level in combination with imaging as well. Because some of the proteins that we're looking at, for instance, DRP1, which is the main fission factor, it's present everywhere in the cell. It's in the cytoplasm, and it's recruited to the mitochondria for fission. So I would not expect the overall level of this protein to be decreased. I would expect it to be present at different places in the cell, and that would
So
you
there is
would
really a
need,
translocation then
yes,
of the protein.
Yes, and you would need a combination.
Irene Hemel, the time appointed for defending your thesis has passed. The degree committee will now withdraw to discuss the quality of your thesis and your defense, and I request that you and your company await the results of our deliberations and our return into this room here. So, the meeting is now adjourned.
The PhD defense has now ended. The degree committee will debate the candidate's performance behind closed doors. This process usually takes about 10 minutes.
Long road to the south side. 7 miles in my rearview mirror, I know what it felt like. My goal's only getting clearer, east side to the west side. No place like home, if it's questions that you've got, go the extra mile and die. On the line, I know you know that I can't read your mind. Years can't fly by now, don't waste all your time. Cause I'll go the extra mile. Long road to the south side, 10 miles in my rearview mirror, I know what it felt like. My goal's only getting clearer, east side to the west side. No place like home, if it's questions that you've got, go the extra mile and die. On the dash, got my laces tied. Long road, I don't waste no time. Break rules because fate decides. With the team and we chase the light. I make a move, fall down, shake it off. I hate to lose, bad branch, break it off. No room for negativity, praise and love. Prepare for deep part cause we're taking off. Take the mileage, traveling so far from home. Gathering knowledge, other connections. I put the time in to get everything I ever hoped for. It's a challenge and we all in. It's a better future that we go for. And even though these nights seem long, I know that I am never on my own. Know you won the battle once you've grown. South side, 10 miles in my rearview mirror, I know what it felt like. My goal's only getting clearer, east side to the west side. No place like home, if it's questions that you've got, go the extra mile and die. Long road to the south side, 10 miles in my rearview mirror, I know what it felt like. My goal's only getting clearer, east side to the west side. No place like home, if it's questions that you've got, go the extra mile and die. Years can't fly by now, don't waste all your time. Cause I'll go the extra mile.
I reopen the session. Irene Hemel, the degree committee here present has assessed the quality of your thesis and your defense. And in view of its positive verdict and taking into account your previous qualifications, the degree committee has decided to grant you the degree of doctor. And Professor Arts is authorized to confer upon you this academic distinction in accordance with Dutch university custom. And I invite your supervisor to now take the floor.
Do you promise to work in accordance with the principles of scientific integrity at all times, to be careful and honest, transparent, independent, and responsible?
Yes, I promise.
By the authority vested in us by law, and in conformity with the decision of the committee here present, I hereby confer upon you, Irene Martina Geertruida Maria Hemel, the degree of doctor, and grant you all the rights attached by custom and law. As evidence of this, I now present you with the degree certificate signed by the rector, the secretary, and the other members of the committee, and affixed with the official seal of the university. Congratulations, Dr. Hemel. I always really enjoy this privilege of addressing you for the first time with your new title, but now I'm going to hand over the floor to your co-promoter, Dr. Mike Gerard.
Yes. Done. Yes? All right, good. Dear Dr. Hemel, I'm the second one who can say that now, so that's good. Irene, let me start by congratulating you on a well-deserved PhD title. I don't know anyone who deserves it more than you do. Now you can let it go and relax. Unless you're worried about the context of what I'm going to talk about. A little bit, okay. Before I go any further, I actually want to aim the congratulations also to your parents who have witnessed really a great achievement of near perfection, as expected, from an amazing daughter that they have raised. Although Irene did all the work, this would not have been possible without the guidance and support whenever needed by your parents. For example, when you were frustrated with your supervisor. Which is a typo. It has to be plural supervisors, especially towards the end of your PhD thesis, I would say. Irene, we have enjoyed an almost 6-year journey in which I saw you develop from a quite shy, kind, but very smart student to a strong, independent scientist that you are now. I know you might struggle with agreeing to the letter, but I have several arguments to change your mind. So first, I want to take you back to the beginning. I apparently still have your email from way back. So on the 18th of April, 2019, at 2:12 p.m., I received an email from you referring to a lecture that you found very interesting, and you were looking for an internship, well, student thesis project, we should call it nowadays. Knowing what I know now about you and sending emails, I wonder how long it took you to write that particular email. We agreed to meet in our old offices, UNS 60, and after a few weeks, I received a message that, and I'm quoting you here, After carefully looking at all the information collectively, I think that the Fission and Fusion project connects best to my interests and way of thinking, and that this project will allow me to grow more than others. I'm not sure about the last part, but you surely grew immensely as a person and as a scientist. You started your thesis project with building a protein-protein interaction network and subsequent validation, which unfortunately was halted through COVID. But we quickly shifted gears, designed a different project in which you checked on the quantification of mitochondrial network morphology, which resulted, I think, in a really beautiful first paper. I was impressed how easily you adapted to the new situation and managed to take the lead, and that was only in your first year. Quite luckily, we were able to offer you a position as research assistant, and to my surprise, you actually accepted that as well. I also have to thank Michelle for that, obviously. Let me see where I am now. So I think I do understand now, after many years, why you chose that position. We, or at least I, had a clear mission to show Ilya that we hired a huge talent for a PhD position. And I tried to obtain funding, but unfortunately was rather unsuccessful. Luckily, you worked so hard that Ilya noticed your talent as well and offered you a PhD position. I still have a huge debt with Ilya that I'm trying to pay back as my role in managing director, but we'll see how that goes over time. Now, second, over time, several experiments, interns, and other sometimes personal challenges were thrown at you. Yet you always were able to adapt, learn new methods, lab and analytics, which you managed flawlessly. And I think your thesis is a really nice example of that. An example, I would hire a bachelor or master student for the project, dump them on you for supervision in the lab, and especially in the beginning, this resulted in quite some frustrations with you, especially one person that I will not name today. I tried to explain to you that you should never compare students with yourself, and I think that was your biggest problem, that you were expecting others to be as excellent as you are and could not accept that others are simply not. But you got aware of that over time, and I think that you clearly, in that part, also grew as a person, as a leader, and were able to really successfully supervise quite a lot of students. Then third, you were a shared PhD student between Michelle and I, which meant you also had to work on the cursed MRI project. And cursed it was, right, Michelle? But the emphasis on was, because you managed to tame that beast, and I think really nicely. You had a lot of bad luck with collecting data, ranging from the scanner updates, crashes, participants that had an accident and had to cancel, and other things. And on top of that, you had to keep 2 supervisors with slightly different interests happy by working both in the lab for molecular biology, which I pushed all the time, but also scanning the subject or your MRI project. Now, we all know that the mitochondria power the brain, so what else is there to say, right? Just kidding, Michelle, without your contribution, which was essential, I think, for the success of this thesis, and I know that you secretly love mitochondria as well, just as much as I like the brain nowadays. In one way, I think Irene actually united us in our research. Now, fourth, which I found very impressive, and it took a lot of effort to get this done, is actually you moved to Finland on your own. I don't think you believed that was possible years ago, that you would do that. I do remember our first trip, actually, to Sevilla, and it was only the second time you would fly somewhere. So I thought, I arranged everything, booked the flight, arranged the hotels and everything. And then when I told you what time we were going to leave, I saw a kind of panic in your eyes, like, so are we leaving that late? So I always go for a flight so that I arrive around 1 hour before the flight leaves. And that was for Irene unacceptable. Because she had heard that the security check might take a while, so she didn't want to risk it. So as a compromise, we actually went way in advance. We arrived 2.5 hours before the flight left on the airport. The security check took like 5, maybe 10 minutes, and we were stuck at the airport for 2.5 hours. I was not amused, I can tell you, because I hate waiting, especially at airports. But if you travel with Irene, there's a big chance that you need to wait at airports anyway, due to either delayed or cancelled flights. What is it with you and airplanes? Almost every flight with you results in cancellation, the flight in Sevilla, or major delays, Umea, and your recent flight actually coming here for your defense. So I have flown a lot and only experienced 3 times a delay. 2 of those were with Irene, while we only had 3 trips together. So I want to give everyone the advice, next time you book a flight, check where Irene is, because if she's the same flight, there's a 50-50 chance you'll have a delay or a cancellation. Despite the early arrival at the airport, I have to admit I really enjoyed our trips together. And these trips also have you made more confident and changed you a bit as well. And you might even have done things on the trips that you normally never do, like crossing the road while the lights are on red. Now the 5th and the final point, almost there. Your people skills or management skills also really vastly improved. As mentioned earlier, you managed to deal with the interns, but also you're a very kind person who's actually really good with people. Everyone in the department enjoys being around you, likes talking to you. And you also realize very quickly, I think maybe after 1 week already, that I'm not a morning person, which makes me actually wonder if you planned this defense so early to get back on me for all the years that I actually annoyed you. Either way, you made sure that there was no meeting, with me at least, planned before 10 a.m. and also made sure that every student knew they should not go to me before 10 a.m. in the morning. I think this changed a bit by now, but that's my opinion. And then also, besides all the work, you also manage the calendar of your colleagues. You managed my calendar almost, because you know when I had meetings, and you prevented Dan, for example, missing certain tutorials by managing his calendar. Now I can go on and on about why I believe you're a great person and scientist, or the good memories that we have of working together, but I guess it's also time to celebrate with your family, friends, and colleagues. I want to finish with telling you it was an honor being your supervisor. To describe what our collaboration means to me, I can only quote a very smart scientist and kind person. There are no words.
Dear Dr. Irene Hemel, also on behalf of Maastricht University, it is my pleasure to congratulate you with the degree you have just obtained. And I would also like to extend my congratulations to your parents and also to the supervisors. So everyone, very well done. And then I would like to thank everyone present here and maybe those who are joining us online. And I would especially like to thank those of the committee who put a bit of effort in preparing nice questions and reading the thesis. And some of you even traveling from quite a distance to be present here. So that's much appreciated by Maastricht University. That brings us to the end of the official part of the meeting, and that leaves me with some household messages. And maybe I switch to Dutch for that. U kunt nu